So in addition to the R groups on a polypeptide backbone, as well as the polypeptide backbone itself, these two major components that are involved in the chemistry of, well, the chemistry of enzymes, there are also molecules known as cofactors. So cofactors are molecules that are covalently or non-covalently bound to an enzyme in order to basically assist in the enzyme's capacity to catalyze a reaction. So there are metals that participate in this. And these metals, your alkali and alkaline earth metals, your sodium, potassium, magnesium, and calcium, are commonly found. And what they do is they are loosely bound and they fulfill structural roles. We also see transition metals. These are going to play key roles in catalysis, and they're very effective electrophiles. So they help out the polypeptide in making some pretty unique chemistry, or not unique chemistry, but chemistry that the enzyme otherwise is incapable of doing. Or the R groups of that enzyme are capable of doing. So basically these cofactors, whether they are organic molecules or metals, they kind of they expand the chemical competency of the polypeptide. So in addition to the presence of metal cofactors or organic cofactors that allow an enzyme to carry out a function it otherwise would be incapable of doing, there are a number of factors that influence enzyme activity. Temperature and pH are two examples. So when you think about pH, I want you to visualize this, not that line, but I want you to visualize a axis that looks like this. On our y-axis, we have activity and enzymes activity. And then on our x-axis, we have pH. And our pH is going to range from, let's give a narrow scope, so from 4 to 9. Now, what this curve is going to look like is it's going to be very low. And we're thinking about this in terms of like a real activity. So an enzyme carrying out an activity, whether or not a reaction is taking place at a pH of about 4, maybe it's not taking place. Hence, this line is pretty much flat. But then it's going to peak. It's going to drop off. So if we go ahead and fill in some numbers here, so 8, 7, 6, 5. Not really drawn to scale very well, but essentially what this shows is that this enzyme has activity within a pH range from about 5.5 to 7.5. Now, if the pH were lower or higher than that range, well, this enzyme's not going to be functional. Now, temperature effect on enzyme activity. Well, human body is about 37 degrees, right? Yeah, about 37 degrees at any given moment. And enzymes are going to, within human cells, are going to be optimal at that temperature. If it's too cold, well, are they going to exhibit some activity? They sure are, but it's going to be lower. Now, this shows a 30, 40, and 50 degrees. What we see is an enzyme's activity at a low temperature is low. As it goes up, as the temperature goes up, activity goes up until it reaches an optimal temperature. An optimal temperature where the activity is highest. If you go above that, it's going to plummet. Something like the hydrogen bonds are going to be broken within that molecule. So basically, this illustrates how an enzyme behaves based on changes in temperature. Now, here we've got a similar graph to what I was showing, but this is illustrating how there are different enzymes that have different activities based on the pH. For instance, pepsin. Pepsin is a protease. That's an enzyme that breaks peptide bonds. It's highly active at a lower pH, so from about pH 1 to about 3. If you go above 3, the activity plummets. Chymotrypsin, on the other hand, if you are in a very acidic environment, chymotrypsin is not going to have much activity. It's going to have higher activity at about pH of 7 here to a pH of about, let's see, that's about 9. So chymotrypsin and pepsin both do the same thing. They are both proteases. They both cleave peptide bonds, but they're going to have different ranges in which they are most active. And why is that? Well, it's because we've got groups that are being protonated and deprotonated at these different pHs. So enzymes can be regulated based on the environment in which they're in, but they can also undergo regulation in a number of different ways. It's easiest, I think, to look at enzymes and their function through the lens of metabolism. So reactions in metabolism are going to take place in a sequence. The breakdown and biosynthesis of biological molecules must be controlled to conserve energy. Metabolism must respond to the environment. For instance, if there's a huge amount of sugar, well, either you want to store that sugar or you want to process that sugar. Now, metabolism is under a total of 4 different major means of control. Genetic control, covalent modification, allosteric regulation, and compartmentalization. So genetic control is, well, in the right environment and in the right conditions, under the right conditions, genes are going to be expressed, enzymes are going to be expressed at the transcriptional level so that they can catalyze a reaction when needed. So enzyme induction, synthesis of enzymes in response to changing metabolic needs. Stimulants can turn on or off genes. We saw that when we were talking about, I believe it was adrenaline. The presence of adrenaline ultimately led to the breakdown of glycogen and therefore liberation of glucose. Enzyme repression, end product of a biochemical pathway, may genetically inhibit the synthesis of a key enzyme in a pathway. So there might be a product that suppresses the expression of a gene that would lead to more of that product. So that would be kind of a negative feedback loop. Now, in addition to genetic control, now when it comes to covalent modification, some enzymes are regulated by reversible interconversion between active and inactive forms. Several covalent modifications cause these changes in function. Phosphorylation, we already talked about that as a post-translational modification. This is going to convert an inactive, or it's going to convert an inactive form of an enzyme to an active form of an enzyme. And that's either phosphorylation or dephosphorylation that's going to do that. Other common modifications, these are post-translational modifications: methylation, acetylation, ubiquitination, and simulation. Now, one that we haven't talked about is this class of molecules known as zymogens. Zymogens are proteases, but the cool thing about them is that they are inactive. So think about it as, well, I can't think of a good analogy, or I haven't thought of a good analogy yet, but these are proteases that they're not functional as they're produced. Oh, actually, heck, here, I've got one. Think about a glow stick. A glow stick is a lot like a zymogen. When you buy a glow stick, is it lit up? No, it's not. It's only lit up whenever you crack it. And you're not breaking it in half, but you're cracking it. You're doing something to it to basically irreversibly make it different from how it started. Now, when it comes to a zymogen is like the glow stick. It can float around and everyone sees, oh yeah, there's a glow stick. It's not doing anything though. When you crack it, now the difference here is that zymogen has to be broken in half. A covalent bond, a peptide bond, has to be broken in order for that enzyme to actually be functional. So whenever you crack it, and in this case we're breaking it in two, then it is actually functional. Now, covalent modification by phosphorylation, one example of that is the enzyme, and we actually talked about this one when we were looking at our previous metabolic pathway. Phosphorylase kinase is an enzyme that is phosphorylated, and what it does is it transfers a phosphate group from ATP onto an enzyme known as glycogen phosphorylase B, producing glycogen phosphorylase A, which is simply the phosphorylated form of glycogen phosphorylase B. So phosphorylase kinase takes ATP, transfers the phosphate group onto glycogen phosphorylase to make glycogen phosphorylase A, and that enzyme right here is then capable of glycogen degradation. Now, allosteric regulation is the form of enzyme regulation where an effector molecule binds to the protein's allosteric site. This causes a conformational change in the enzyme. So an activator is an effector that makes the substrate binding much easier. And so what that's going to do is it's going to lower this Km. Now Km is a unique term for an enzyme's basically the affinity that it has for affinity. It's a measure of the affinity for the substrate. So a low Km, this is inverse, or this sounds backwards, but a low Km means high affinity. A high Km, therefore, means low affinity. Okay, so an activator is going to lower Km. What this is also going to do is it's going to increase the Vmax. What is a Vmax? That is the maximum velocity of the enzyme. So the fastest that this enzyme can catalyze its reaction is, well, a higher Vmax. Now an inhibitor, let me erase some of my writing here from a minute ago. An inhibitor is going to do a couple of things, because what it's going to do is it's going to increase the Km. So it's going to decrease the enzyme's affinity for its natural substrate. It's making the substrate binding harder. And it's also going to decrease the Vmax. So it's going to make it harder to bind the substrate, but it's also going to slow the enzyme's reaction down. And what these are going to be able to do is they're going to be able to respond to certain intracellular changes or intracellular conditions within the cell. Now, the last thing that I wanted to talk about was compartmentalization. So this is our last layer of control of metabolism. Different processes occur in different organelles. So ATP synthesis, for instance, takes place within the mitochondria. Protein synthesis takes place in the cytosol. ATP breakdown takes place in the cytosol as well. So you're going to produce this ATP in the mitochondria so that it can be utilized. Compartmentalization allows for better control of processes, keeping these processes separately. You don't want synthesis of a fatty acid to take place in the same place that you are breaking down a fatty acid. So you have them segregated from one another so that the fatty acids can be broken down, energy can be extracted, and the fatty acids can be built up in another place so that they're not immediately broken down. In addition to that, this compartmentalization also enables something like the lysosome to function naturally. So the lysosome is an organelle that has a very low pH, a pH of about 5.5. So you can keep lysosomal proteins separate from proteins elsewhere in the cell. Well, I guess that kind of wraps up what I wanted to talk about for our first part of enzymes in chapter number 7. We'll continue this discussion in chapter number 8. All right, thank you very much. Have a good one.